When we were looking at the record prior to the launch, NASA sent all of the experimenters a variety of samples to test, as I mentioned. There was a sample from the Antarctic taken from high in the dry valleys that Chris mentioned that the GCMS examined and reported that it failed to detect any organic matter. Years later, when I was browsing the literature files, I found that a classic chemical analysis made at the same time by the same team found indeed there was 0.03% organics present in that sample. And I found just last week, this thing never ends, I kept looking and I found that not only was there organic carbon present, but there was 0.003% organic nitrogen, a matter which has never been addressed in discussing these Viking findings. Well, as I delved into the files, I also found that at the same time that the GCMS was unable to detect organic material in Antarctic number 726, we had run 726. I pulled the record out and we had detected. There are some puzzling facts that we would like to look at further with respect to the GCMS. It's the only thing we have, the GCMS, to compare to the LR results to try and rationalize them. What I have just shown does this in one sense. In other words, it is possible that on Mars, the labeled release got a response, but the GCMS did not, because this response came from too few organisms for the GCMS to see. And in that sense, the result could be, the two results could be perfectly compatible. Looking at the GCMS in the next view graph, there are some caveats in accepting those results. First of all, there was no telltale, no sensor saying that this tiny oven, 600 cubic millimeters, was indeed filled. And one of the ovens was not run because they had a feeling that it might not be filled, but this was more intuitive than anything else because nothing told them that it was filled or wasn't filled. The values of the abundances, the experimenter said, may deviate from the actual values by a factor of 2 or more. We're talking about very small abundances of the material seen. Then there's a statement, those subsystems involved in the soil analysis, namely the sample ovens, tubing, and valving prior to and possibly after the gas chromatograph column, were less clean in one lander ovens than the other. For this reason, the interpretation of the soil analysis requires subtraction in a qualitative and quantitative sense of background data acquired during a blank run performed during cruising through space. The blank was obtained on oven 2, but you have to assume that oven 3 was the same, and you subtract based on that assumption. Then, for the only two analytes detected, CO2 and water, it is stated by the experimenters that several factors severely complicate the determination of the amount of water. As a matter of fact, the amount of water is not directly determined by the instrument, but indirectly by the way that one of the regulators in the instrument operated. So they're pointing out that in the very worst, we could be off by a factor of 5. Well, if you apply a factor of 5 to the results, none of the results with respect to water or carbon dioxide, which are the only two results that indicate that a sample was ever in the instrument, are significant. Now, look at the next view graph to explore this hydrogen peroxide thing. Well, this shows the only molecule, I'm back at the GCMS, the only molecules detected were water and carbon dioxide. How much water was detected? It's 0.01 to 1 milligram. So had this 1 milligram and the CO2 50 to 700 parts per million CO2. Had those not been there, or if they are not significant, there's no evidence that the GCMS got a sample, which would certainly make its interpretation different with respect to the LR experiment. There is also the problem that looking at these small amounts of these molecules can be difficult because, remember, in between each of the runs, the GCMS was sitting on Mars surrounded by carbon dioxide in the atmosphere, and it has been pointed out, frosted on the surface of Mars so that a speck of water or ice somehow, if it gained entry, could produce results that would make us wonder. I'd like to move to considerations about the next obstacle to accepting, next view, life on Mars, and it's the one that, beg your pardon, I'm a slide ahead of myself. There are reasons that constrain the peroxide on Mars, and the most important reasons are the two equations. In the atmosphere of Mars, it's true that water is dissociated and that products under the influence of ultraviolet light then produce hydrogen peroxide. On the other hand, hydrogen peroxide, as anyone knows, as a kid having put some on a cut, pulls it out of a brown bottle, because light, especially ultraviolet light, destroys it so readily. If you look at the rate coefficients, hydrogen peroxide is formed is destroyed 10 million times as fast as it's formed. So you would think that it would have a difficult time raining down onto the surface of Mars before being destroyed by the ultraviolet light.